Okay, so let's look at DNA transcription. So we're still in the central dogma of molecular biology as it was explained in our previous topic, which is DNA replication. So we're still looking at DNA. So today I'll not focus on the replication of DNA. So we have to produce RNA. Then at a later stage, we have to produce a protein. So this is the central dogma of molecular biology. So the information from DNA to RNA has to be transcribed, and this process is what we call transcription. And this is our focus for today. This is what we want to look at. How does this process happen? How does the information change from DNA to RNA? And what is the reason why this information has to be changed from DNA to RNA? This is what this video is all about. So from RNA, the information here will have to be translated. So this will be our next topic. In our next videos, we'll be looking at translation now, how a particular protein is produced and how the information from RNA is translated to produce a protein. So what I have here, this is the central dogma of molecular biology. So our focus for today, as I've already mentioned, it's here, transcription. So transcription of DNA. So what is transcription? So transcription, this is the process by which a copy of DNA is changed into RNA. So you have got DNA, then DNA is going to give us RNA. Now what do we know about these two molecules? So DNA, we know that there is a double helical molecule, a double-stranded molecule, so we have something like this. Whereas RNA, this is a single-stranded molecule. So what we are going to, this process of transcription, you have got a DNA. So this DNA, the information from DNA has to be changed. That change from DNA to RNA is what we call transcription. So even if DNA has got two strands, the newly produced RNA only has got a single strand. So we're going to produce a single strand from this particular DNA. So the reason why this information has to be changed from DNA to RNA is simple. As we know that DNA this is the molecule which stores genetic information. So the genetic information that is stored by DNA has to produce a protein, and this DNA cannot move out of the nucleus, but the information has to move out of the nucleus to the cytoplasm. That is with respect to eukaryotic cells. So now, meaning that this information now has to be changed in a way that it has to be the same. The information has to be the same, but it has to be changed in mRNA. Okay? So this process is what we call transcription, how the information is changed from DNA to RNA. So that's what we want to look at today. So we're going to look at transcription in prokaryotic cells and eukaryotic cells. So prokaryotic cells, then we also have got eukaryotic cells. So just as in DNA replication, we said that DNA replication in prokaryotic cell and eukaryotic cell is quite similar, but when it comes to this process transcription, there are a lot of differences on how transcription takes place in eukaryotic cells or in prokaryotic cells and how DNA transcription takes place in eukaryotic cells. So for that matter, I'm going to cover DNA transcription in two separate videos. That will be DNA transcription in prokaryotic cells and DNA transcription in eukaryotic cells, so that you can get to understand transcription in prokaryotic cells on its own and eukaryotic cells on its own. Okay, but in this first video, I'm going to give you the overall description of this same process of DNA transcription. Okay, so as I've already said, as I've already said, that transcription is the process by which information from DNA is changed into RNA. So we know that we have got DNA is a double-stranded molecule. So if I have two strands here of DNA, I've got that strand, another strand here. These are two strands. These two strands of DNA, they run in an opposite direction. So let's say if this is a 5' end, 3' end, this is a 3' end, then I have this is a 5' end. Now we are saying that the molecule which we are going to produce, which is mRNA, is a single-stranded molecule, meaning that out of these two strands, it is only one strand which will act as a template. So what I have there in white, this is acting as a mRNA. So what I have here, this is my DNA, a double-stranded molecule. Then what I have down here, this is going to be now messenger RNA. Before I get into the details here, we know that we have got three different types of RNA. We have got ribosomal RNA, we have got transfer RNA, then we also have got messenger RNA. All of these are the three different types of RNA. So now, what you have to know is that all of these different types of RNA, they all come from DNA. They all come from DNA. But the major type of RNA here, which is going to be discussed much, is this messenger RNA. Messenger meaning that it's carrying the message. So the message that is this RNA is carrying is the message which comes from DNA. So we're going to have a sequence of bases on this DNA. That sequence of bases is going to be specific for a particular protein. So we call a particular sequence of bases on DNA as a gene. So a gene is going to be made up of a sequence of bases. So those sequence of bases, they are the ones now which they have to be transcribed into RNA. And this type of RNA is going to be the messenger RNA. Then later on, this messenger RNA is going to give us a specific protein about that gene. So at the end, we are going to have protein. Meaning that as we move from RNA to protein here, this is where we have got the process of translation. Okay? But for today, we just want to understand how do we move from DNA to RNA here. That's all. So basically, all the three different types of RNA, they all come from DNA, as we are going to understand what really happened there. So this RNA that we are going to discuss much is going to be this one, messenger RNA. But I also talk about ribosomal RNA, transfer RNA, and messenger RNA. So now what I was saying here is that DNA is a double helical molecule, so it has got these two strands. So between these two strands, we have to produce only a single strand of RNA. We have to produce only a single strand of RNA. Okay? So meaning that we have between these two strands of DNA, one of them has to act as a template. A template is a guide. A template is a molecule which is going to have complementary bases to this strand or to this molecule which we want to produce, which we call messenger RNA. So let me restart. So I have two strands of DNA. Mind you cannot have RNA from without. You have to have DNA. So that is my double helical molecule of DNA on top there. Let's give directions. So this I'll label this as 5' end. This is 3' end. This is a 3' end. This is a 5' end. Why I'm giving directions here is because direction matters. Just as we saw in DNA transcription or replication. So we have to produce a single strand of RNA from these two strands of DNA. So we're saying that between these two strands of DNA, one of them has to act as a template. A template is going to be a strand that is going to have complementary bases to this RNA. So what I have down here, this is RNA. Whereas what I have here, this is my DNA, a double helical strand of DNA. So the strand which is going to have complementary bases to this strand here is what we call a template. So the template strand, between these two strands of DNA, one of them acts as a template. So the template is the strand of DNA which runs in the direction 3 to 5, always, and not the one which runs in the 5 to 3. Not this one. Okay? So not this one. This is the template. So this strand of DNA is the one which has got complementary bases to the molecule of RNA which we are going to produce. So if we look at these two strands which I have here, the one which is in 3 to 5 direction is the strand down here. So this strand, we call it as a template. So this is the template. So that's a template. With respect to the strand which runs in 5 to 3 direction, this strand is going to be non-template. Because the other one is template, automatically this strand is known as non-template strand. So that's a non-template strand. So these two strands here, they are given several names. So one of them is we have got template, the other one is a non-template. The template strand, this is the strand which is going to have complementary bases to our RNA here which we want to produce. So another name of template strand is what is known as, so let me give you different names of these two strands. So I have, we are saying that the 3 to 5 strand, I'll first give names to these ones. Then I also have got the 5 to 3 strand. This is with respect to the strand of DNA. So these strands which run in the direction 3 to 5, we call it as a template. So it has got complementary bases to the RNA which we want to produce. It's also known as the nonsense strand. It's also known as the non-coding strand. Non-coding strand is also known as the minus strand. Okay, so all of these different names, they all mean the template strand. Then another name is known as the antisense. Nonsense is the same as antisense, so let me also include that. It's also known as the antisense. I'll explain what all these mean. Then when it comes to the strand of DNA which runs in the direction 5 to 3, that is this strand on top there. This strand is what is known as the non-template strand. So it's non-template with respect to the template strand. So this is non-template, also known as the coding strand. The coding strand. Another name is sense strand. Then what else? So this strand is now the plus strand. Okay, so these are different names which are given to these different strands which we have here. So now one might get confused, like we are calling this strand which runs in the direction 3 to 5 as a template strand. At the same time, we are saying it's a nonsense, antisense, or non-coding. What does it mean? So what it means here is that the strand which we call the coding strand, the one which runs in the 5 to 3 direction, this strand here, this is the strand which gives specifications about the protein to be produced. Whereas this strand which we call the template, this is just going to contain the information about the coding strand. So one thing which you are going to observe is that the bases, the sequence on the sense strand, they are going to be the same as those on the RNA. So let's say here, if I had bases on the coding strand, let's say I'll start with ATG, then ACC, AUC. Sorry, we can't have uracil in DNA. So here, let me say TCG. So if these are the bases on the, if this is the base sequence on the sense strand, what are going to be the complementary bases on the template strand? So the complementary bases on the template strand of DNA down here, let's give those bases, base sequence. So here we know that we're going to have A, then there, no T, then AC, then I'll have this will be T, then I'll have GG, T, then A, G, and C. These are going to be the base sequences on the template strand. So now these bases, they have to be complementary because this is a DNA. These two strands are complementary to each other. Even their bases, they have to be complementary to each other. So in short, these are going to be the base sequences which we are going to have with respect to the bases that I've given on the coding strand. Now what we are saying here is that this strand RNA here to be produced, let's first give direction. So the direction of the RNA which is going to be produced here, it always runs in the direction 5' end to 3' end, and nothing in reverse. 3 to 5, this is wrong. So RNA is only in the direction 5' end to 3' end. That's all. So you're not going to find RNA in this direction. That's not possible, because RNA is a single-stranded molecule. So its direction is only in this direction, 5 to 3. Okay? So the direction on the RNA here, which is going to be produced, is always going to be in the direction 5 to 3' end. So the direction here is going in this direction. Okay? So now we are saying that this strand is complementary to the template strand. If you look at the template strand, its direction is the 3' end to 5' end. Automatically, this is telling us now that the strand to be produced, because it's complementary to the template strand, it has to have the opposite direction. This is the reason why RNA will always run in the direction 5 to 3 and not in a reverse. So now let's see the bases which we are going to have here. You have to take note that in RNA, we have got bases uracil. You will not find uracil in DNA. You are going to find thymine in a DNA. Okay? So let's see how do we get these base sequences. So what I want to give here now are the base sequences on RNA with respect to the bases that I have on DNA here. Because this is going to be a gene that is going to be transcribed with respect to that. So now the base sequences on the RNA here, they are going to be complementary to the bases on the template strand. So we start with, we have got thymine there. We are starting with thymine. So thymine is complementary to which base? Adenine. So here we're going to start with adenine. Let me use a different color. So okay. So we're going to start with adenine. We have got thymine. Thymine is complementary to adenine. Next is adenine there. Adenine is complementary to which base? This is RNA, so we can't have thymine here. RNA, we cannot have thymine there. So adenine will be complementary to uracil with respect to RNA. We have cytosine. Cytosine will always pair with guanine. So this is going to be our first codon on the mRNA. Then we come to, we have got thymine there. So where there is thymine, we are going to have what? Adenine. Then guanine will have cytosine. There is a thymine there. What are we going to have? We are going to have adenine. Then adenine, we are going to have uracil now. Then C, G. This is going to be the base sequence on mRNA. Now I want you to observe one thing here. If you look at these base sequences, let's pick the first three on the mRNA. So the first three bases, we have got A, U, G. What's their direction? Their direction, we have got 5' end here, and we have got 3' end here. Now compare these bases which we have here with the first three bases on the sense strand of DNA, or the coding strand of DNA. So I'll pick the first three. The first three I've got ATG. So I've got ATG. Then the direction 3' end and 5' end. If you look at these bases, what can you notice? Where there is adenine, you also have got adenine in RNA. Where there is uracil, where there is uracil is replaced with or where there is thymine in DNA, because the strand down here is the one for DNA. This is the sense strand of DNA. So where there is a thymine in DNA, you have uracil and RNA. Where there is guanine, you're also going to find the same guanine. So one thing which I want you to observe here is the base sequence between the RNA produced and the base sequences on the coding strand. So the reason why we call this as the sense strand or the coding strand is because it has got the same codons or base sequences as the base sequences on the RNA, except that where there is thymine in the coding sequence of DNA, you are going to find the uracil. So this is what I want you to understand as we get into this topic. Okay. So if you was given a strand of DNA like this one here, then we are being asked to state the base sequences of RNA. Simply, the base sequences on the RNA, they have to be complementary to the base sequences of this strand of DNA, which runs in the direction 3 to 5. Okay? If you don't want to do that, then just use the coding strand. The base sequences which you have, the base sequences of DNA on the coding strand that you have, just change where there is a thymine, put uracil, then maintain everything. What you have now, then you have base sequences on the RNA. So, get to understand these two strands: the template, non-template, coding, non-coding, sense, and the non-sense. So it's not making sense, it's just keeping the information. Hence, it is the antisense or non-sense. This is the one which codes for each and every molecule or protein to be produced. So the information about every gene or molecule protein to be produced, they are coded by that strand there. That's why we call it the coding strand. Okay? So this is what we have to achieve at the end of the day. We have to produce a single-stranded RNA molecule. So now, what also do you have to understand apart from you understanding the sequences or these bases or the name of the strand? So,